Bony fish evolution into tetrapods. Brought to you by me, Logan Howerton.
Me, Corinne Gutman.
And me, Brian Thompson.
Around 385 to 360 million years ago, at the end of the Devonian period, bony fishes began to evolve into tetrapods. The first image you see is the picture of a bony fish. The second one is the picture of a tetrapod.
Tetrapods are four-limbed animals and constitute the class of Tetrapoda. Class Astopithecus, bony fishes, includes the largest number of living species of all classes of vertebrates, with more than 28,000 species. They share several distinguishable features: a skeleton of bones, scales, paired fins, and one pair of gill openings, jaws, and paired nostrils. Living Astopithecus are further divided into two subclasses.
Actinopterygii.
Ray fin fishes is a clade of bony fishes that generally lack the fleshy base to paired fins and possess air sacs that normally function as a swim bladder.
Sarcopterygii.
Typically referred to as lobefin fishes or lung fishes, usually possess paired fins with a fleshy base over a bony skeleton, a notochord, and two dorsal fins. Lungfish specifically are thought to be the closest relatives to tetrapods because lungfish are air breathers, which is a characteristic shared with terrestrial vertebrates.
While most vertebrates share similar characteristics, bony fishes have some other unique structures compared to tetrapods. Their jawbones usually consist of adductor and abductor muscles responsible for jaw movement. These muscles are in their pelvic fins as well. Lungfish specifically could survive on land, considering that they can breathe air and use their pelvic fins to propel themselves around. Many species of bony fishes also have a gas-filled bladder called a swim bladder, which serves principally in maintaining neutral buoyancy. The swim bladder was developed initially as an organ for respiration, proven by the lungs that lungfish have developed. Lungfish's gills alone cannot keep them supplied with enough oxygen to live. Therefore, they swallow air to fill up in an air sac or lung. The air sac is surrounded by veins that bring blood to be oxygenated.
The lungs. The lungs are an essential connection in the evolution of lungfish to tetrapods. Surfactants are lipoproteins that cover the surface of the lungs and are vital for pulmonary function. Lungfish have experienced increased surfactant genes that are very similar to tetrapods and are 2 to 3 times higher than other bony fishes. This increase could indicate an adaptation in lungfish favoring air breathing. The expression of SHH, a vital regulator of lung development, has been studied. It was found that SHH was heavily expressed in developing the lungs and envisioning the development of the right-sided lung, similar to amphibians.
Clement and Long's study pertains to the air-breathing adaptation in Devonian lungfish. This air-breathing adaptation evolved independently from tetrapods, and this suggests divergent evolution. Divergent evolution is a process where organisms closely related independently evolve different traits due to having to adapt to different environments or ecological niches. Acquired traits make them unique and distinguishable from tetrapods, and this is likely because of similar environmental pressures.
Fin to limb. Michael Coates compares Hox genes expressions in fins and tetrapod limbs. Development of zebrafish pectoral fin bud and tetrapod limb are quite distinguishable. Pectoral fin growth stops earlier, so they are smaller. As growth ceases, cell reproduction finishes, and the end of its fin's outer skin transform into a protruding fold, enclosing the developing dermal rays. Dermal rays are bony structures derived from intramembranous bone formation, and they form components of the vertebrate skeleton: skull, jaws, gill covers, etc.
The composition of the zebrafish HOX-D gene complex appears to resemble that of tetrapods, with posterior members expressed similarly. HOX-A expression is similar but less complex than that of tetrapods. The posteriorly restricted HOX-D expression in limbs and fins provides evidence for long-established theories of homology between the posterior fin in bony fish and the proximal joint of tetrapod limbs. we can conclude that bony fish's lineage led to tetrapods.
Bony fish can also be compared to humans, and air-breathing lungs and fins resembling tetrapodal limbs and parallel development all correlate with this connection. And studies on bony fish can be applied to humans because of all the comparisons that they have. Hyperostotic bone in marine fishes has many attributes of human cellular bone, and these fish bones can be used to compare the effects of fracturing hyperostotic and normal bones, which may lead to new approaches to skeletal deficiencies.
Hyperostotic growth in fishes was once presumed to be related to fungal growth or cancerous bone tumors and can be linked to genetic and ecological factors. For example, 5 oarfish stranded on the California coast. More active fishes were more likely to have hyperostotic bones. Certain environmental conditions determine the bone density of the fish. Most fishes with prevalent hyperostosis were found in tropical, shallow water conditions.
Comparable to fish, genetic and developmental disorders can drastically affect the skeleton. Osteogenesis imperfecta, common disorder, patients have a mutation that interferes with the body's collagen production. Lack of collagen production can lead to dental problems, hearing loss, severe bone deformity, and even stillbirth. Physical activity, diet, and absorption of minerals, vitamin D, can also determine the bone health of humans. How can knowledge of bony fishes be applied to humans?
We have found due to research that because of the commonalities between humans and fishes, that using fish as a smaller scale, we can test the fish with certain drugs or certain treatments that could possibly soothe the effects of the osteogenesis imperfecta in humans, and we can use the fish to find out new things about disorders in humans and to try and prevent them or just decrease the symptoms from these genetic disorders.